welcome to the April 2026 MIDAS webinar. It's my pleasure to welcome Dr. Rebecca Wattam from the University of Virginia. Dr. Wattam is an associate professor and the outreach lead for the Bacterial and Viral Bioinformatics Resource Center, which we're going to hear about in just a little bit. Dr. Wattam's work focuses on developing and delivering accessible bioinformatics resources for the study of infectious diseases, with particular emphasis on antimicrobial resistance and comparative genomics. And she's led over more than 80 hands-on workshops worldwide and is creator of the Coursera course Bacterial Bioinformatics, which has reached tens and thousands of learners. And for anybody who's taken one of those courses, you know that's a lot of work to put that together. I first met Dr. Wattam through an introduction from a colleague at University of Virginia back in the fall, and we have been told by our steering committee for MIDAS and others that we need to do a better job of helping people understand how they might use sequence information and genomic information in their infectious disease modeling. And so that's what we're going to hear about today. So with that, Dr. Wattam, I'm going to pass it over to you. We're going to take questions. If any come in as we go, please let us know. Otherwise, Rebecca, looking forward to hearing what you have to say.
Okay, great. Thank you for inviting me. Is everybody at Pittsburgh? I assume it's a larger resource.
Yeah, no, this could be, folks could be coming from just about anywhere.
Okay, good. Okay. So what I plan to do today is give you an overview of the resource by slides, and then go into a live demo of the things we offer on the resource. I know that you guys are most interested probably in the data, but I can't help myself talking about the other things in the resource that mean a lot to me, like the bioinformatics services and things like that. So I'll also be going over that. So let's see if there we go. So the BRCs were originally established in 2004. This came on the heels of the anthrax attacks in 2001. And the first target, they were established for infectious disease researchers, more with an urgency of making those people aware of the data, giving them tools to study the data. And for a long time, we were told targets, drug targets, vaccine targets, interventions, preventions was the scale of it at the beginning. We integrate genomic, omic, and metadata. And since 2015, at least our BV-BRC has been providing analysis tools and visualizations. So for a long time, the resource was all about the data and viewing the data and accessing the data and bringing in more and more data. And then in 2015, it shifted more to analyzing that data. And as you well know, that data has exploded. When we first started the resource, it was called Patrick at the very beginning in the 2004 round, and we were bacteria only. We only had like 8 bacterial genomes, and now there are millions. So the BRCs also support large scale and user-driven workflows, and it enables researchers that are, let's say, computationally naive with the ability to analyze, do some very complex analyses using the resource. But we also enable people who are computationally literate. They can come in and sweep the data and whatever they want. So we've been funded for 22 years. We have more than 77,000 registered users, and per month they submit 20,000 or more jobs across them all. And like I said at the beginning, we had 8 bacterial genomes in 2004. Now there are 1.3 million genomes in the resource for bacteria, 15 million for viruses. And anybody who signs up for the resource all gets access. It's free to sign up for, you get a private workspace. And as our users have grown, that workspace has grown, and now it takes up 820 terabytes of space. And it's all housed at Argonne National Labs through the University of Chicago. So the users keep growing, the citations keep growing. And when you look at our user community, we see a lot of everybody. They're microbiologists, they're epidemiologists, they're bioinformaticians and data sciences. It spans the government and state workers coming from the different labs, the different institutions, NIH, NIAID, CDC. We also get some people from cancer, even energy, because especially where the bacteria are concerned with the viruses, we are just bringing in data that are from pathogenic viruses. But I guess you would think that all viruses are pathogenic. So let's say pathogenic to humans. But for the bacteria and the archaea, we bring in everything because we want people to be able to make comparisons. So we get, people from industry using it. We have a lot of people who are making yogurts and probiotics, and they're coming in and using the data. We had a really interesting group the other day contacting us. They're trying to make probiotics for people who are being treated for cancer, and they needed to be sure that the bacteria that they were using did not have any antimicrobial resistant genes, because you don't want to, feed a person who's immunocompromised, give them a bacterium that can beat out an antibiotic. So it's amazing to me often the breadth of people who are using the resource. There were some people in Italy a few years ago that were trying to engineer bricks that were for building, that were hollow on the inside with fluid that had bacteria who could do photosynthesis to power the building that they were trying to build. It's just amazing. All the downstream applications that can occur with this data and the kind of people you run into. So some of the key features of it, and let's talk about the data and the metadata. We go to GenBank every night and we bring in new genomes. So if it's in GenBank and it's a genome, a bacterial genome specifically, but also the targeted viral genomes, we bring it in. And once it's in, we re-annotate it. And I'll talk about that in a minute. We also have a team that scours the literature because there's a huge push by our team for understanding antimicrobial resistance. So they're looking at the literature for these large studies that are looking at, hundreds or thousands of bacterial isolates, and they're exposing them to a whole panel of antimicrobials, and they're recording the susceptibility and resistance and the MIC values for those. Now that data, because it's so intense, and you have to put data, if it's funded by the federal government, you have to put it in a public resource. So it's stored in the Sequence Read archive, and that's the only place it exists, or only place it did exist. So once we read these papers, we go in, we grab that data, we assemble and annotate those genomes, and then we run machine learning on them to try to identify classifiers that are associated not only with susceptibility and resistance, but also with different MIC values, so that when someone brings an isolate in that matches the species of these larger studies, it will give you a prediction and tell you whether we think it's susceptible to amoxicillin or resistant to amoxicillin based on what we have run with our machine learning algorithms.
Okay. Rebecca, quick question. Since
Sure.
this is not really a bacteria crowd, could you just define what you mean by when you say MIC value?
Oh, minimum inhibitory concentration. When you're culturing bacteria in the lab, you put a little bit of antibiotic in it, and what they'll do is they'll plate it out at increasing concentrations of antibiotic. And so you'll see at what point the organism can't grow anymore. And so that's what they're talking about with that. So being able to predict at what concentration you could kill this bacterium, that's what those studies are aimed at.
Thank you.
Also, when the data comes in, we get out the metadata associated with it. And there are 90 different facets, but some of them are the host, the location, like the country, the city, isolation information, like if it was isolated from blood or cerebral spinal fluid or whatever, the date it was collected, the country. When we first started the resource and had those 8 genomes, the genome sequence was the most valuable thing because it was so rare. Now that we have so much data, really it's the metadata that's so valuable, because by filtering on the metadata, as you can see below, and I'll show you later for different levels, you can open up these filters and quickly go through the data and collect data that you're interested in. That's the most valuable thing, to show you things that you want to compare to your data, or you just want to grab data. And you could easily grab something like all the Streptococcus pneumoniae genomes that were isolated from blood in Egypt in 2023. I don't know if we have any of those, but if anything meets that particular set of criteria. But that's one of the things you could do when you have the metadata tagged to it. And where do we get that metadata? From the biosample, from the notes associated with the GenBank Association, and from the bioproject. These are things that when researchers submit data to public archives, they are required to fill out and provide that sort of information. So it's a great service to all the communities when they do this, because it makes easier for the rest of us to do these comparisons. So we also have other data like viral epitopes, protein structures. I've been using the protein structures so much because I've been analyzing resistant and susceptible bacteria and looking at the differences where those different single nucleotide polymorphisms, called SNPs, where they occur in these proteins of interest. And then I look at that on a structure, and sometimes you can have structures, they've taken snapshots really of these proteins that are bound to a antibiotic. And then you can see if the places that are changing in the bacterial gene are near where the protein is interacting with the antibiotic. So I use that a lot. We also have a lot of curated omic data sets looking at differential expression, and that's what you see in the figure of up and down regulation depending upon this different phenotype of these organisms. We also have microbial and host protein interactions, surveillance records, and serology records. So we keep trying to bring in data that is useful for our community. So when we get this data from GenBank, or bring in the genomes from SRA or the reads from SRA and assemble them, we take everything through specific annotation pipelines. Because we keep the old, if they had previous annotations, we keep that for people to see that. But we also re-annotate things so that there is an overall, so that you can make a comparison between things, and you're not comparing across different annotation systems. It's sort of, we're trying to make everything apples to apples so that you can see that everything has been done the same way. For all the bacteria and all the archaea, we use RAS-TK, and there's the pipeline for it up there in the upper left. Bacteriophages, for those of you who don't know what those are little viruses that actually attack bacteria. And the United States went full on into antibiotics in the 50s, but other countries, Russia, Georgia, and Brazil, they've kept bacteriophages active in that research so that you treat a bacterial infection, especially one that's not responding to antibiotics, with a virus that will, and I'm sure maybe you've heard about the people who've are dying of C. diff and they treat them with a phage and bring them back. So anyhow, that's just an aside story. We have the Phanotape pipeline for bacteriophages, and while we don't do all viruses, we use Vigor4 and also one called Peptide for the different viral annotations. So I talked briefly about the antimicrobial resistant annotation, and this describes some of it, but I wanted to point out the things that you get if you have an organism, and these are some of them in the left-hand panel, that we have the data where we can predict, or we will predict that it will be susceptible or resistant. And some of these things we can predict MIC, minimum inhibitory concentration value as well. And so when you run it we have our annotation service that automatically does this. If you come in with the Streptococcus pneumoniae genome, it's going to give you a prediction. And if you use any of these on the left-hand side, you're going to get a prediction of susceptibility or resistant, and even the other thing, depending upon what you use. And so when you get that, it'll give you the prediction that you can see in that first panel under, I'm afraid to do this because sometimes it skips it ahead. It'll say, this is what we predict your genome will be susceptible or resistant to. It'll also show the region and the areas around it that you see in the next little panel. And it will also give you a list of the antimicrobial resistant genes that we identified and the strength of the hit to those. So for a while now, we've been going full burn on antimicrobial resistance because it's an increasing problem in the United States and globally. So let's turn to tools and services. To me, this is the meat and potato of things. Right now, we offer, when we first started launching bioinformatics services in 2015, we had 5 services: assembly, annotation, RNA-seq, proteome comparison, and a little differential expression tool that we used. So now we have 33. And with these things that we provide reports, we provide tables, and we provide visualizations to help enable people to see the differences in their data. So it's a really powerful resource. And these are just some of the visualizations that are included in the resource. And I look at a lot of papers that cite the resource, and these things are often showing up because we offer the visualizations too, and you can download them in a scaled vector graph or an SVG and use them in your publication. So it's pretty sweet. So is anybody using these services? They certainly are. I've summed these by year, and I didn't include 2026 because we're not into there yet. But you can see that it started slow and it's picked up. 2021 was a bumper crop year. We were hot in COVID then, right? So maybe that was it. But I do know that someone went in and got every single bacteriophage known to man and reannotated them all and then took them away and analyzed them themselves. So we know what happened in that particular year. So I've just listed the 10 most popular by year, and then all the other remaining services are binned at the end. But the powerhouse is really annotation and assembly. That's where people are really coming and doing a lot of their analysis work. Also BLAST. Now, taxonomic classification we launched pretty recently, and that's come on pretty strongly. We launched it in 2020. So that's been a very successful service because it lets people see what's in their data, especially since we're getting more and more mixed samples and mixed data. And we're providing people with ways to not only find out what's in there, but also pull out the genomes. We have a metagenomic binning service that pulls whole genomes out of mixed samples. So it's pretty sweet. And I use that a lot too. I actually use all of these a lot. I had mentioned earlier that if you register for the resource, you get a workspace. It's a private workspace. You can put whatever you want in there. And right now it has no limits. We also have shared workspaces so that you can collaborate with teams and also classes, which we have people that do. And your data that's in there is seamlessly integrated, although only you can see it, with the public data. So if you want to have your data and run phylogenetic trees or do a SNP analysis, you can easily do that. And it's really great because it's there for you, and it's really hard to do those kind of things, especially if you're computationally challenged. So this has really been very powerful for a lot of our researchers. Now, this might be of interest to most of you guys, is that we have both programmatic and batch access. We have a command line interface. And so everything that I talk about, every search that I show you, all the data I show you, you can easily get that through the command line interface. And you can launch the jobs as well. I don't know if that's as interesting to you guys as the data, but you can use the command line interface to get the data. And in fact, people seem to like the way that we have the data and the structured metadata, and they come to us probably even more than GenBank to grab that data, and they take it away and do whatever it is they do with it. We just know about it because we see it, that they've referenced us in their publications. A lot of the machine learning stuff, people are coming in and accessing the data for machine learning. So we have a lot of help documentation. Oops, I don't know why that happened. We have tutorials, we have reference guide. I make so many YouTube videos. I am so tired of hearing my own voice because a lot of those videos are me describing how to do everything. So we have that, and we also have the help desk. So in 2026, I just wanted to briefly tell you some of the new things that we have. One of these is we're launching this AI Copilot. It's in the beta form right now that helps you understand, get access to, interpret things that are in the resource. So we have a team working on that. The dream is, and we've been playing with this, we actually did it at a codathon, is I gave it, although you can't do this right now in beta, but hopefully by the end of the year, this is something we've been playing with. I gave it a couple of papers that I'm interested in and said something like, here's my private data. Here are these papers, plot out an analysis for me. And I've always been sort of a naysayer, I must say, on the AI. And I was told to start using ChatGPT for the project back when it started. And I was not a fan then, and it's gotten so much better. But for our own thing, it gave an analysis. I think it was like along the lines of what maybe a graduate student would do, in that it wasn't like it gave something that was feasible. It wasn't the way I would have done it, but it had me pause and think about it and think, yeah, I think you can do it that way. That's an interesting way to do it. So I must admit, I've gone from being a total skeptic to being much more impressed with our own Copilot, and of course with what I see online as well. We have new services. I don't know if these are too interesting to you guys, but we have these are for the viruses. We combined with the viral community in the fourth round. Before that, we were Patrick, which was all bacterial. And then there was Viper and IRD, which were the two viral bioinformatics resource centers. And then NIAID said, we're only going to fund two, one for bacteria and viruses, and one for eukaryotic pathogens and insect vectors. And so we combined with the viral people in one. And so we've been making a push lately on the viral services. So we have a subspecies classification service so that researchers can bring their data in and say, what is this? And they've included some new measles and mumps and into the service. So it helps you figure out where it is within that species. Viral genome and assembly workflows. You would think that bacteria, well, we're all humans and our genomes are gigantic, but compared to a virus, a bacterial is really large. And you'd think that viral genome assembly, how complicated can it be? Well, it's kind of complicated. So it's been a while for us to really get good at this. So the genome assembly service, we've launched that. Many of the BV-BRC group is in, the primary people are in Chicago at Argonne National Lab. I'm at the University of Virginia. We have a group at the University of Virginia. Some of the viral people are at JCVI in California and Washington, and at the University of Alabama our co-PI is there. Our PI is in Chicago. And several of these groups work with epidemiologists and CDC, the CDC project called PGCOE. And so because we're working with epidemiologists, we're also developing tools that they can use. And then we bring them also into BV-BRC. So the SARS-CoV-2 wastewater analysis service is one of those. We also have the sequence feature variant type service. This is for viruses. And the docking service, which is pretty fun. You can give it the structure of something that you want to see how it docks, how it interfaces with a particular protein, and you can explore that using the docking service. So that one's been fun too. Another new service, this is just for bacteria, is the core genome MLST. I do a lot of comparative genomics. I must admit, I don't use MLST very much. I don't understand why people like so much to distill an entire genome when you've got the whole thing there, distill it down to 20 genes and make assumptions about that. But bacteriologists love it. And so we brought that in. And so it computes a core genome. And it's only right now for 32 different species. And it can be used for surveillance, outbreak detection, and population studies. And you notice I have a tool listed here, Chewbacca. One of the things we like to do is we're always following the literature. We don't want to reinvent the wheel. If there's a great tool out there that the community is using and that they love, we bring it in and incorporate it into the resource and give them credit and tell people to cite them. So that for us is a win-win rather than us redoing everything that somebody has already done successfully. And also along that lines, when things get antiquated, we bring in something new and retire the old service that we had. Another thing we brought in is whole genome SNP analysis. Now we had a SNP analysis for reads, sequencing reads, where we could run that against a reference genome and it would identify SNPs. This is not with reads, it's with assembled genomes. I can remember really wanting to do this about 15 years ago to examine the vaccine strains to see where the SNPs were compared to the progenitor strain that they had come from. And we just didn't have that service because I didn't have the reads for those things. It was all old Sanger data. But I had the context. So this is a new way you can do it. The tool is KSNP4. It's a reference-free approach. So you give it a set of genomes and it runs it and just summarizes the differences for you. And it gives you, identifies the SNPs, and it also generates a phylogenetic tree. So another thing we've worked on recently is any genome that's annotated in the resource, we black them, or now this is an HMM approach, against specialty databases that do things like antimicrobial resistance, virulence factors, drug targets, transporters. So that we've been doing that for a long time now. So now we're trying to do more of that for antimicrobial resistance gene prediction. Right now, we just updated AMR Finder Plus and CARD from the Comprehensive Antibiotic Resistance Database. They call it RIG CARD, but it's CARD. That's the database out of Canada, MacArthur's database. We also have updated the virulence factor database, brought in the new ones for that, the therapeutic target database, and the transporter classification database. Something brand new is BAC-MET to identify metal-resistant genes. These are really important in bacteria and a potential source of treatment, of knocking out a bacterial pathogen. So those are important ones to have. And also we've expanded our machine learning models for susceptibility and resistance and for MIC. One thing that we've, in this round of the proposal, this fifth round, is education was important. It was called out that we needed to do. So we brought in a specific group that is trying to interface with people who are educators at the college level and potentially moving into high school with those. So it's great. What we're trying to do is make it available to everybody and have lessons and stuff that educators can use. So the first year, we just had a webinar about from certain speakers that were using the resource. And it was surprising how many people joined that, excuse me, that were using the BV-BRC for teaching that we have no idea about across the world. So a lot of people are using it, and now they know how they can create a workspace for their classes, put in lesson plans, put in exercises, share jobs and data with classes and have them do a lesson, do a semester worth of work. And I can usually tell when people, because sometimes I'm in charge of checking the help desk, and I know when I'm getting a lot of things from people who are from the same university coming in that they're having problems. Something's going on. They don't understand something in the class and they're all asking the same question. So I can tell when it's getting close to finals time somewhere because I get those things. So we're soon going to have an educator website session so that they can all be there and work on that. And we hope to host future education meetups. We also, now this might be interesting to you, we also, oops, I don't know why it's doing that. We also do outbreak tracking, mainly viral outbreaks. We provide the overview, we give them phylogenetic trees, they have quick access to all the genomic data associated with that. And we also provide genomic data maps so that they can see, like we show down here at this one, where the H5N1 cases were predominantly in the 2024-2025 flu season. We also have surveillance data. You can go and search for it, grab the maps, and you can see where the isolates were located from. And you'll notice that this one is a kind of funny picture. It's a migratory path. Some of these maps have the migratory path of birds because often birds are transmitting this transmit. It's all data to me, but they're actually living organisms, and the birds are bringing them from one area to another when they are doing their migratory patterns. So that's a kind of, well, I was going to say that's a kind of fun way to look at the data, but actually it's not fun if you're in that area and getting the latest flu virus, and who knows how bad it's going to be. So what I thought I'd do now is exit and do a demo. But before I do that, I guess I'll give you guys the slides so that you can see these things, but you can follow us on LinkedIn and Slack. And I also want to, these are the sites that we all work at. So I just wanted to give credit to everybody that's associated with the project. So now I want to go into the demo. So let's see if I have it up here. Okay. Let me make it big. Okay. Any questions before I start?
Not seeing any, but thank you.
Okay. So let me move this bar over a bit so I can see these things. And I'll make it a tad bit bigger. So across the top, this is the landing page for the resource. And here we have a global search, and here you have ways to jump to the different data. But I rarely come to this page, except I come down here and go to the YouTube channel. That's generally the way I find it, is going that way. But the main working part of the resource is here in these tabs across the top. And because you guys, my understanding is that you're interested in more data, I want to step through the data that we have here. So I can click here on organisms, and it opens this drop-down box. And you can see we have bacterial pathogens, viral families, and featured viruses. And then here we have all bacteria, all archaea, all viruses, and eukaryotic hosts. Now, when I've given these workshops, people have said, I can't use your resource. And I say, why not? I say, you don't have my bacterium. And I say, oh, yes, we do, because we have the pathogens, because the BRCs was originally set up as a pathogen, pathogen-centric. These are the predominant pathogens, but you can use the toolbox, the global search, to quickly get access to anything that you want. So it's always a little slow when it's going through Zoom, but that was pretty fast. So you can see that I can get to here. It'll show me the taxa, the genomes, the features, all, and it goes on and on, the epitopes, protein structures. But if I clicked on Streptococcus pneumoniae, I've been doing a lot of work with Streptococcus pneumoniae, so that's what you're going to hear today. And you notice that this has a bunch of tabs too. And note that right now we have more than 32,000 Streptococcus pneumoniae genomes. And that's a lot of data. And imagine if you're a researcher trying to decide, what am I going to look at? How am I going to, I can't build a phylogenetic tree with 32,000 genomes. So I've got to pare it down a bit. So I'm going to click on the genomes tab here. And I'm going to click on the filters here. And so you could quickly go in and say, I want to see all the good genomes isolated from Australia in 2003, if that's what I wanted. And there's more metadata. If I were to click here, these are all the different metadata facets that I could filter on here. So it's just that kind of quickness. I could also do something like, let's say I wanted blood. Say I was interested in things that were isolated from blood. And you could see that I've got 12,253 results. Now, each of these, blood is mentioned somewhere. So if I pick on one, select one, and you notice when I click on it, not only does the green action bar come up with icons of things I can do with it, but I could go down here and try to see where it is talking about blood and if it's talking about blood somewhere in here. And I could also click here and add, hold on, I always like to add the isolation comments. Put that in and oops, sometimes I have to bring it over so I can see it. Well, this one doesn't seem to have that, but let's see if I can do isolation source. So you can see that there's a lot of stuff that you can access here. Okay. So when I put in the isolation source, you can see that it's blood. And you can see like here, it's blood from 60 years, probably a 60-year-old person, or maybe the blood had been stored for 60 years. So just being able to grab things that quickly, it's pretty amazing. And I don't know what kind of analyses you guys do, but this is the kind of stuff I do all the time, and it really helps me with my work. So I also wanted to talk about AMR phenotypes. When I'm going to quickly go back to the home page. This is showing all the genomes across these 32,000 genomes that are, let's see, I want to order them by count, that have antimicrobial resistant prediction data. And so I could go into AMR phenotypes here to see that data. And once again, I can click on the filter and I can say, I want to see, let's see, I want to see penicillin. And look at the kind of data we have here. You can see it from data that we have actually established laboratory methods for susceptible and resistance, and also the minimal inhibitory concentration. So, and these are the papers that are associated with that. So I could see it, say, I want to see all the resistant ones, and I'm interested in broth dilution. So I can get those. There are 2,065. I could collect them all and create a genome group of that to examine later. So that's just some of the kind of things you can do with that data. Here are the proteins. I've been doing a lot of work on antimicrobial resistant. And if I want to see all, whoops, didn't type in there. If I want to see all the TET-M proteins, this is the protein that's important in the resistant to tetracyclines, I can grab them easily that way. Protein structures, this has been so much fun for me. So if I'm looking for a particular protein, and then you can even see, there's one I really like in here. So let me see. Actually, I'm going to do oxacillin. Okay. So I could click on that, say I want to see the structure. It'll take a second to load. And you can see the protein structure, and you see when I highlight the ligand, that's where the antibiotic is bound to it. There is the pink is the antibiotic. The other is this protein that's very important that the antibiotic targets, and we have different views of it too. So you can twist it around, and there's the antibiotic sitting in this pocket of this protein. Let me go back to the old thing. I don't want to cover those so much, but I want to go back to the home page and I want to show you that this is a taxon landing page. So the whole thing about BV-BRC is piling up data and integrating data at different levels. So this is at the level of a taxon. We also have data at the level of a genome, and it can be a public genome or it can be a private genome that you brought in. And here we have resistant and susceptible predictions from our machine learning algorithms. It tells us the taxonomy. It gives us an MLST. It gives us links back to the original data because we don't want people to come here and get totally lost or have it be disconnected with the other data resources. We're trying to interconnect to everything. It tells where it was sequenced, all this information about it. Here are these genes that are important in either virulence or resistance or whatever. So I can get the AMR phenotype for the genome. You notice we have all these tabs across the top where you're filtering down on it. I can get a circular view. This is very popular among people, showing me the forward strands and the different genes of interest, AMR genes, virulence factors, transporters, and drug targets. I'll get the genome browser. Let's see if it loads quickly, because I know I'm yammering on as I have a tendency to do. And probably this isn't too much interesting for you guys, but you can zoom in oh, you know what? I'm going to zoom out just a bit more. Okay. This is just fortuitous. It's showing me the result of some of the machine learning algorithms we've run on this thing. This particular region of the genome has been associated with the resistance to trimazazole. I'm used to saying sulfamethoxazole, trimethoprim, and sulfamethoxazole. So you can increase it here and even see what that region is and the 6' translation. You have to admit, even if you don't do data or if you're not interested in this, this is kind of cool that you can do this. So we have the proteins, the protein structures, and the specialty genes. And for this organism, I can say, I want to see all the antibiotic. Oh, it's been doing this funny thing with this. So anyhow, the filter but I could be able to filter on everything using this. And I could also click on the properties and filter data that way. Okay, so that's pretty much, let's go to proteins here. So we've looked at data accumulated at a taxon, at a genome, and we also accumulate data at a particular gene. So this is a gene or protein, and I click on the feature icon, and here we have yet more tabs and yet more data associated with it. So see, there's this huge amount of data in the resource, and we're trying to structure it and make it so, because you can't put everything on one page, but it builds on itself. I think of it as like nested dolls, and now we're at the nested doll for the proteins themselves. And it shows you who's around them, the protein families, what the genes are, and we have the browser and the compare region viewer. I'll click on it briefly. Sometimes it takes a while to load, but this is a way that you can explore gene neighborhoods. Okay, I'm just going to leave it because I know that we don't have too much time. And while I don't, oh, look, isn't that pretty? So it's showing you the gene you came in on, and it's looking for similarities across the gene neighborhoods around that. So we also have ways to search for the data. For example, if I wanted to quickly find ERM-B as a gene that's important in macrolide resistance, and I could quickly look here, and it'll show me all the genomes that have this particular gene. So 19,000. See, that was pretty fast. So we have different searches to allow you to filter the data. Oops. We have tools and services. Let's just click on genome annotation. So I can annotate anything. I have the recipes that I use. So these are the pipelines that are launched. And up here, this is a direct link to the user facts. These are tutorials and videos. And well, not quite finally, we have the workspaces where I can see my home workspace. I could see the workspaces that I've shared with other people. So my home workspace is very busy because I've been using it for years now. The workspaces I share with others are shown here, but here's the BV-BRC workshop is the one that I teach off of. And I just wanted to point out that if you go into AMR workshop, Streptococcus, and introduction, this PDF, there's a PDF file here that steps you through basically this live demo I've been showing you. But I also wanted to show you the kind of things that you can put in a workspace to share with collaborators, like PDFs, like slides. Ugh, I shouldn't tell you videos, not videos. I'm the only one who gets to use videos because they take up so much space. Okay. And then finally, if you have questions, here are tutorials. And if you want to talk to us, you contact us here. And so I will leave I won't leave, but I'm opening it up because I see I've been yammering on for quite a while. And are there any questions, anything that I can help you with, or do you all just need to go to the nearest bar and have a stiff drink and try to forget about everything you've heard?
No, thanks. That's really interesting. Do we have any questions? I've got a couple things I could fire away with if nobody else has anything. So I guess my question for you, what I think might be most interesting to folks here is to think about uh, the infectious disease outbreaks that we might see, viruses, for example, and also like which viruses are where and when do we see patterns that might go into models that we might use? Because obviously it's one thing to say, well, there's an outbreak in this location at this time, but if we think that there's a new variant coming up, that could be something we throw into a model.
Mhm.
Obviously, looking at data like yours is one way to think about when might those things interact, or even building models of predicting where the next variant might come up or what it might look like. So could you speak to data we
Well,
might use
I
for those
would,
sorts of things?
we have tons of data for SARS. Let's just see how much data we have for SARS. 9 million genomes for SARS. And if you go in and look at them, and it takes a long time for 9 million genomes to load, but you could filter the data. It's going to take a while to load. You can filter it, but you could all do this through command line, which I'm sure that most of your community can do easily. And you can say, I want to see everything from 2021 compared to 2022. I want to look at the SNP variations. All of that is things that you can do as we wait for this to load. Let's try something a little simpler. Zika might be easier to load that. That's only got 2,000 genomes. There we go. So along those lines is you can see where the data is coming from, and you can see that a lot of the data is in these years. So you can grab it depending on the country or the data, and then you could run through the different pipelines, the SNP pipelines, whatever it is you want. But what I'm assuming, it seems to me that most of the public health people that I interact with, they want to use their own tools. I mean, computer scientists, I love you guys, but you always want to redo what is already there because you always have more confidence in your own ability than anybody else's, which is fine. But my point is that most of the epidemiologists are coming in and I want to grab all the complete genomes from this thing that were from 2016 and just grab all of that. And then I can easily get all the results and take it away and have my way with that data. Is that along the lines that you're talking about, Harry?
Yeah, sort of. I mean, I'm speculating here. I don't have a specific use case in mind, but I think that this is a set of questions that somebody might use genomic information for when they're building infectious disease models. So I'm throwing out a sort of a hypothetical here. Uh, but I think that's sort of really the point here being that you could come up with all these different variants.
And you could,
And
like the data
right,
is tagged now
right.
by the metadata
Hmm.
that you have, that is, you've decided on for some reason. So you've grabbed that specifically, this is your interest and this is what you want to look at it. And once you get that, then you can start manipulating that data and run whatever machine learning or whatever it is you're interested in doing with it. That's what I think, especially for the epidemiological crowd, is this is the easiest
Yeah,
way to
yeah.
grab it. Doing something similar at GenBank is not easy to get the data
No.
in the same way. So this is a good way for, I think, your community to get that data.
Yeah, absolutely. Absolutely. And so a related thing to this, you mentioned a command line interface, and maybe that's what this is for, but uh, would the command line interface be the tool that I would use if I wanted to, say, automate a search like this? Or
Well, I wouldn't use it because I am one of the I'm computationally challenged, is what I like to say. So
yeah.
I'm very good at using the resource and filtering in, but everybody else I works with and all the people who are coming up now, they are all using the command line interface. So
Mhm.
you go up into help, you go up into command line interface, and here it's got the installation and tells you exactly how to do it. And you can also get the pipelines too and run them on your own computer, but
Mhm.
why do that
Well, why would
when
you do
you,
that?
yeah,
Sure.
because you can get the run time is free at Argonne National Lab. And so if you have problems with it and you're not able to figure out how to do something,
Mhm.
then you go back into the contact us here and you say what the problem is you have. And the guy who designed it is sitting there, and I assign that ticket to him, and he responds to you and says, "This is the command line that you need to do to
Mhm.
get this
Mhm.
data that you want."
Mhm.
And so that's how easy it can be.
Got it. Thank you. That's really useful. Okay, we're about at the top of the hour. Do we have any other questions here? I think there might be one Q&A here. Okay, so um, I don't have any other questions right now, unless anybody else give one more second here. Before we go, I do just want to mention one brief sort of comment that I'd like to make, if I might share my screen for just a
Oh,
minute.
sure. Let me stop
Um,
sharing.
so hold on a second here. I got to get ready to do this. Um, and this is sort of a community announcement for or a comment for those of us in the MIDAS world. That I want to take a second to hold on a second as I do this right. I'm trying to do two things at once here that I haven't done lately. Um, it's not working, but all right, let me just do this. I wanted to share a short remembrance of Wilbert Van Panuys. Um, Wilbert was the director of MIDAS from 2019 to 2021. Before that, well, at the same time, really, he was also an assistant professor and then associate professor of epidemiology here at the University of Pittsburgh. Uh, and he was responsible for the creation of the TICO data set, which was a large data set where they transcribed records about infectious diseases worldwide for over 100 years, and that's still available at tico.bit.edu. And It's now run by the MIDAS Center under our leadership. Wilbert is also why I am here. Literally, he brought me in to work with the MIDAS Center in 2020. And in 2021, he took a position at the National Institutes of Allergies and Infectious Diseases there as the director of their Office of Data Science and Emerging Technologies. Wilbert was diagnosed with the glioblastoma sometime around 3 years ago. He died on April 13th. And so I just want to take that minute to remember him. He was a real leader in data science for infectious disease modeling and a real force of energy for that and an enthusiastic advocate for the work. And he will be missed. So thank you for indulging me on that. And thanks to everybody who worked with Wilbert, and our condolences go out to you. So with that, I will say thank you, Rebecca, for the talk. Really very interesting. I encourage people to take a look at the BV-BRC. It's a great resource. And thank you. Take care.
Thank you.